Watch this crab. He's a model parent. He cleans the eggs under his abdomen, fans them with oxygenated water, helps the larvae escape when they're ready. He does everything a mother crab would do. He isn't a mother, and those aren't his children. The animal under his abdomen is not an egg mass. It is another animal, Sacculina carcini, a parasitic barnacle, yes, a barnacle, that has done something to this crab that no virus, no fungus, no microbe has ever done quite like this. It dissolved itself to get inside him. It grew through every organ in his body. It removed his sex. It made his body grow a female's shape. And then it rewrote his nervous system to raise its children as if they were his own. He does not know any of this happened. By the end of this video, you will understand exactly how, and you will understand why it isn't an accident. It is evolution doing something with no mind that vertebrates can barely do with one. Let's start with how she got in. In the cold, plankton-rich waters of the North Atlantic and the Mediterranean, anywhere European green crabs gather along rocky shorelines, there is something drifting that nobody notices. A microscopic larva, so small the human eye cannot pick it out from any other speck of drifting life. It is called a cypress, a swimming larva that looks more like a tiny shrimp than what it will become. It has antennae. It has paired swimming legs. It has compound eyes. What it does not have is the rest of its life. Because in a few days, almost everything you just saw will be gone. Sacculina carcini is a barnacle, a real one, member of the same group that builds the white encrustations on harbor pilings, on the hulls of ships, on the backs of whales. Filter feeders, sessile, harmless. But somewhere in its evolutionary past, a lineage of barnacles took a different turn. Instead of cementing itself to a rock and filtering plankton from the passing current, it found another solution. It found a way to live inside something else. When this transition happened, somewhere in the deep Mesozoic, perhaps earlier, there were probably intermediate forms. Barnacles that attached to crab shells the way external parasites still do today. Then barnacles that pierced the shell to feed on tissue beneath. Then barnacles that grew deeper, abandoning the shell entirely. We do not have the fossil record to trace it step by step because the soft internal phase doesn't fossilize. But the order Rhizocephala, containing roughly 300 known species across multiple host taxa, is what survives of that long inward migration. Sacculina carcini is one of the most refined. To get there, the larva will pay a price almost no other animal pays. The female cypress swims until she finds a crab. Not just any crab, a specific kind. Carcinus menis, the European green crab, is her favorite. How does she find one? Through chemistry. Crabs leak compounds into the water, amino acids from their urine, traces of molting hormones, specific proteins from their shells. Carcinus menis has a distinctive chemical fingerprint, and the cypress is tuned to it. Other crabs swim past unrecognized. The cypress ignores them. Her entire larval life is a few-week-long search for one species' breath in the water. When she finds one, she lands on its leg, specifically on a fine hair-like sensory bristle at the base of a leg called a seta. Why a seta? Because that is the only point on a crab's body where the cuticle, the hard armor, has a thin spot. The seta is essentially a soft opening in the crab's defenses, no wider than a few microns. Through that opening, Sacculina can reach the inside of the crab. But she cannot go through it. Not yet. Not while she still has a body. So, she gets rid of one. Over the course of about a day, she undergoes the most extreme metamorphosis in the animal kingdom. Her shell dissolves. Her swimming legs reabsorb. Her muscles, her digestive tract, her sensory system, all of it. She throws away almost everything that made her a barnacle in the first place. What remains is a tiny cellular mass with one hollow needle. This stage is called the kentragon, root bearer. It is a syringe. It is a delivery system. It is what the female now is. She gave up her body to become a way in. And then she is inside. He does not feel any of this. A seta is a sensory bristle, useful for detecting current, vibration, water chemistry. But the place where the cuticle thins to allow it is too small to contain the kind of nociceptors that would register an injury. There is no pain pathway for what just happened. There is no biological alarm. From the crab's perspective, an insignificant detail at the base of one of his legs has shifted by a micron, nothing more. He goes on with his day. What is now inside the crab is a cellular mass with no organs, no senses, and no shape. The crab cannot feel it. It would not react if it could. But over the next few months, that mass is going to grow, and it will not grow like anything else that has ever lived inside a crab. What enters a crab through that injection is not a parasite as we usually picture one. It is a seed. There is no head, no body, no organs, just a small clump of cells with one job: to grow. And it grows in a way nothing else inside a crab grows. It grows roots. Not all at once. In the first 2 weeks after injection, the cellular mass is barely larger than a grain of rice. By the end of the first month, it has extended its first major rhizoids, branching outward in 2 or 3 directions through the body cavity. By month 3, the network reaches the major internal organs. By month 6, every region of the crab's body has been threaded with parasite tissue. The name rhizocephala literally means root head. When biologists first dissected an infected crab in the 19th century, they didn't believe what they were seeing. It looked less like a parasite and more like a plant that had taken root inside the host. Branches, tendrils, roots reaching through every system of the crab's body. First, the roots find the hepatopancreas, the crab's liver-like organ where it stores energy. They wrap around it, they penetrate it, and they begin drinking. The crab keeps eating. The crab keeps living. Most of what the crab now consumes is feeding the roots inside it. You might expect the crab's immune system to detect this. Crabs do have an immune response. They produce phagocytic cells that wrap around and neutralize foreign tissue. But Sacculina's roots are thin, distributed, and constantly extending. They do not present a single target. They look, to the crab's body, less like an infection and more like a slowly growing extension of the host's own tissue. The immune system fires nothing. Then the roots find the gonads. Whatever the crab was doing reproductively, it stops. The gonads atrophy under the root's presence. Tissue thins. Cells stop producing eggs or sperm. The crab is castrated, not surgically, not with a knife, cell by cell, over weeks. And And this is what makes Sacculina one of the most thorough body snatchers ever documented. The roots find the nervous system. Sacculina's rhizoids extend along the crab's central nerve cord, into the ganglia, around the optic stalks, into the muscle bundles that control movement, into the small abdominal nerves that control the most precise movements a crab is capable of. The parasite is not just inside the crab. It is inside the crab's wiring. This phase has a name biologists use almost reluctantly: the interna phase, the internal phase, the phase where, from the outside, the crab looks completely normal. It scuttles between rocks, it hunts, it eats, it avoids predators. It looks like every other green crab in the tide pool. But it has stopped being only a crab. And there is one more thing happening, slowly, during these months. Something the crab cannot do anymore, that it doesn't know it cannot do. It cannot molt. The leading hypothesis for why is endocrine, molt-inducing hormone, produced by the crab's own Y organ, drives the cycle of shedding and regrowth that every healthy crab depends on. Sacculina's roots, settled in the right tissue, appear to suppress that hormone signal. The crab's body is told, chemically, that no molt is necessary. That command repeats indefinitely. The crab cannot shed its old shell. It cannot regenerate a lost claw. It cannot continue along the normal arc of crab development. It is frozen, physically frozen at the size and shape it was when the parasite first arrived. Time for this crab has stopped. And then, one day, somewhere between 6 months and a year after the injection, the parasite is finally ready. She pushes herself out. The thing that was injected as a microscopic cellular mass that grew silently through every organ of this animal has built itself a body again, just not the body it started with. The sac hanging beneath the crab's abdomen is called the externa, the visible adult body of the female Sacculina. Everything else about her, the network of roots that destroyed the crab's reproduction, the tendrils reaching into the nervous system, is still inside the host. The externa is the public face of an animal that is otherwise entirely hidden, and the position it hangs from is not arbitrary. A female Carcinus minus carries her eggs under her abdomen, tucked into a wide brood chamber formed by the abdominal flap. To carry that egg mass, her abdomen must be wide. Female crabs have wide abdomens for exactly this reason. Male crabs have narrow abdomens. They don't carry eggs. They don't need the platform. Sacculina needs the platform. And in some places, Sacculina has been remarkably successful at building those platforms. In endemic Atlantic and Mediterranean populations, infection rates of 30 to 50% are not unusual. Half the crabs you'd find on certain stretches of European coast are carrying a parasite that has reshaped their reproductive biology. An entire generation of crabs that should be feeding, growing, mating, and producing their own young turned quietly into nurseries for somebody else's children. On the most affected shorelines, the parasite has effectively become the dominant strategy for crab reproduction. Just not crab reproduction by crabs. So in a male host crab, over the months of the interna phase, the parasite's roots have not only modified gonads and nerves, they have modified body shape. His abdomen has widened, his ventral plating has reshaped. The triangle of a male's abdomen has slowly become the broad, rounded crescent of a female's. He still looks like a male crab from above. His claws are still male-sized. His coloration is still male. But the part of his body that mattered to Sacculina, the underside, is no longer male anatomy. It has been remade into a brood platform. He has been physically feminized. And he does not know. But there is another parallel story happening. One nobody talks about because it is so small. Once the externa is out, it begins to release a signal, a chemical scent that attracts the second character in Sacculina's reproduction, the male. The male Sacculina is also a cypress. He arrives at the externa, finds the pore, and enters. And then he does what she did when she first arrived at the crab. He gets rid of his body. His shell, his legs, his eyes, his muscles, everything. Within hours, he is nothing but a small cellular mass that produces sperm. He has no body to speak of. He never will. The male Sacculina never has an adult body. He lives the rest of his life as a sperm pack inside the female. 2 reductions. The female sacrificed her body to enter the crab. The male sacrificed his to enter the female. What makes this strange isn't that one sex reduced, it's that both did. Evolution arrived at the same answer. Get smaller, faster, more single-purpose, twice, in the same species, from opposite directions. The female by getting inside another animal, the male by getting inside her. Sacculina is one of the few species where being an adult has been almost completely outsourced, for the female to the host, for the male to the female. What remains in both cases is just the part that matters for the next generation. And now everything is in place. The crab is castrated, feminized, cannot molt, carries a foreign body where its own young would be. The parasite has eggs being fertilized by males she will keep with her for the rest of her life. All that's left is the part that, from the outside, will look like the most natural thing in the world. He is about to become a parent. But the parasite isn't only inside his body. By now, it is inside his behavior. And what it makes him do next is the strangest thing of all. Because for this to work, Sacculina needs the crab to perform a sequence of behaviors a male crab has never performed in his entire life, and to perform them perfectly. Inside the externa, Sacculina's eggs are developing, tens of thousands of them. Each will become a cypress larva. Each will need to escape into the open sea at the right moment to find the next crab. But the externa cannot release those larvae by itself. It hangs from the underside of the crab. It is anchored there. It has no muscles of its own to push the eggs out. It has no way to fan oxygen across them as they develop. It has no way to time the release for tides, for currents, for the moment when survival is most likely. It needs a parent. And it has one. The crab. What follows is something that, if you didn't know the biology, you would never suspect. The crab's pleopods, the small leg-like swimming structures on the underside of the abdomen, begin to fan, rhythmically, steadily, pushing fresh oxygenated water across the externa. This is what a female crab does for her own eggs. The same motion, the same rhythm, for someone else's brood. When debris settles on the externa, the crab grooms it off, using its claws the way a female would clean her own developing eggs. When the externa needs to be repositioned, the crab adjusts it. And when the time comes, when the cyprids inside are ready, when the tides are right, when temperature and salinity have aligned, the crab does something a male crab has no biological reason to know how to do. He helps the larvae escape. He fans them out. He pulses his abdomen at exactly the right rhythm to release them at exactly the right moment. He does what a mother crab does for her own children. And in his entire life as a male, he was never supposed to be able to do any of this. He shouldn't even know how. So how does he? The honest answer is that we are still working it out. The leading hypothesis goes like this. The crab's brain, and crabs do not have a brain in the way we do, they have ganglia that perform the work of one, already contains the wiring for parental behavior. Every female Carcinus minus is born with a neural circuit that runs the maternal program: fan eggs, clean eggs, release at the right moment. The circuit exists. It usually fires only when the crab has eggs of her own. Sacculina's roots sit directly on top of that circuit. We do not yet know exactly what signal the parasite sends to switch the circuit on. Hormonal mimicry is suspected, chemical signals that imitate the hormones a normal egg mass would produce, fooling the crab's nervous system into believing eggs are present. Tactile mimicry probably plays a role. The externa is physically shaped like a clutch of eggs, hanging in the exact right position. The roots in the nervous system may also fire neurons directly, simulating the signal a mother crab would send to herself. Likely all three at once, and the parasite, which has no brain, does not have to know any of this is happening. It does not plan, it does not choose. The roots grew where they grew. The chemicals are released in the patterns evolution stabilized over thousands of generations. The host's existing parental program runs. Sacculina did not write the program. The crab already had it. The parasite only had to learn how to flip the switch. Mechanical reflexes on the parasite side, genuine maternal behavior on the host's. The two meet at the externa. What looks like a mother caring for her child is, from the parasite's point of view, a hijacked program running in a hijacked nervous system. There is no caring. There is no mother. There is only the right chemical at the right time on the right neuron, repeated for as long as the externa keeps producing eggs. The crab does not know. And he will go on doing this, fanning, cleaning, helping the next brood escape for the rest of his life. And this is why Sacculina carcini unsettles us in a way few other parasites do. Because what it shows us is that parental behavior, the most intimate, the most evolutionarily ancient act associated with caring for one's young, can be reproduced by something with no mind at all. Given enough time, given enough generations, given a small enough pressure on each step, evolution does not need to think to nurture. In an earlier video, we met a parasite that performs surgery without a brain. We called it the surgeon without a mind. Sacculina carcini is its counterpart, the mother without a mind. Different organ, same horror. Somewhere in the cold North Atlantic, right now, a male crab is fanning a foreign body with the gentleness of a mother fanning her own children. He will keep doing it until he dies. He will never know what he is. If you're the kind of person who likes peeking behind the curtain of our eerie, beautiful world, the kind who wants to know what evolution looks like when it works without thinking, then this channel exists for you. The deep dives keep coming. The stranger ones haven't even started yet. Subscribe so the next one finds you. There is more ahead. Parasites that hijack brains directly. Fungi that perform their own kind of takeover And next time, we leave the parasites behind and look at something even simpler. Not an animal, not a fungus, not even technically alive. A virus that taught the world what a zombie looks like thousands of years before any movie did. It has been with humans for thousands of years, and it does all of it without ever having a face. You will want to see this one.